In last session today, we are going to talk about various techniques, different tools involved for doing protein research, and of course, looking at even complex proteome analysis, uh, which is going to start from some very classical techniques, which I'm sure you have used at some level, especially electrophoretic techniques or the chromatographic techniques. But then we'll also take you to some of the more complex and more newer versions of the instruments, especially different type of array-based platforms or different type of mass spectrometry platforms, how they can be used for generating big omic data sets. And of course, together only by having the mixture of the classical as well as the more high throughput technology, you can do a variety of research. So in this session, we are going to focus on chromatographic methods briefly, electrophoretic method, and then move on to mass spectrometry, followed by microarrays and SPR. So given that the first two are already quite a bit known, chromatography and electrophoretic, I will have less emphasis on that and maybe only brief overview. But as I go along, then I will talk in more detail about other technologies. So what we are talking right now, the technologies which can help us to study the complex protein mixtures in the field of proteomics. And sometime our goal is to identify the new proteins or biomarkers which could be indicative of a given disease or given disorder, given problems. That's really big hunt right now for in this pandemic time. So we are looking at identity of a given protein or looking at quantification of a given protein, healthy versus disease kind of case. Or we also want to identify the function of a given protein, where this protein is getting expressed, what type of assays we can develop to identify those protein, to characterize the protein, understand its function. Those comes under the functional proteomics. So to do this kind of technologies, with the proteomics, one field is known as discovery proteomics or abundance-based proteomics, which essentially looks for what is the change or the abundance of a given molecule. And many times we also look at a lot of image processing type of data generated from these when you are looking at how the signal intensity is obtained from control data point for the given gene or protein versus from the disease individual, how that intensity is getting changed, or how you can see the spectral count is getting changed. So again, many of these things require a lot of data analytical tools, which is really part of this entire field. Discovery proteomics could be based on different type of electrophoretic method like the SDSPAGE gel or two-dimensional electrophoresis, followed by mass spectrometry to identify these proteins of interest. Functional proteomics, which now you already have identified some proteins of interest, but you do not know what the protein do in the system, what is their function. So to identify that, then you look at other approaches like protein arrays or surface plasmon resonance. Some of these can do protein-protein interaction studies or protein-drug interaction studies. Also identify that what is the strength of the binding, and those could give some idea and clue for the functionality of the protein, and that comes under more of the gel-free high throughput approaches. So a typical or sometime the standard workflow of proteomics, especially if you do not have very high throughput setup, that involves around that you can take a chemical, any biochemical technique. You want to separate your complex protein mixture, and then you want to resolve those on the gels like shown here, 2D gels. For each protein which is shown here, which is a spot, you want to identify what these proteins are. You want to quantify those signals from control versus disease conditions, do bioinformatics searches, and then identify the proteins and do more of the structure function analysis. So this is one of the workflows, but of course there are many workflows which is one could use. I'm just trying to give you a simplified view and picture of how to get started in this area. But let me go back a little bit and talk about some basic biochemical techniques, and then we talk more about the proteomics technologies. So chromatography is one of the real backbone in the all the separation sciences when you want to really separate the complex mixtures. Even I just showed you the nano LC, nano liquid chromatography, or ultra high pressure liquid chromatography. So in any of those, you're going to use these columns, which contains some material which is packed like C18 material in this case, and then you are going to separate your peptides from that particular column. So that was the case which we just used, but This chromatography can be used for a variety of molecular separations. So material which is used to pack the column is called the resin or the matrix, which is usually some small beads which are having certain chemical groups attached to them. And how they are going to bind or interact with the proteins, that also determines the nature of the chromatography. So there are different type of principles involved of separating these protein mixtures or peptide mixtures using different type of chromatography. So we'll just take first the gel filtration chromatography, which is very basic when you are really going to look at the principle of size separation. In this case, the beads are made from the polyacrylamide, dextran, or agarose. And what you're looking at when you're passing the protein mixtures from the chromatographic resin, then based on the size, they're getting separated. So the small molecule, like sometime very small molecule protein or even the salt, they will be retained in these beads. So they are going to take longer time to travel. The one which are larger size, they may not stay in the bead, so they have to travel outside of those. So they will move fast. So larger protein molecules will elute first, followed by medium size, and then the small ones are retained. Then you can elute them out. So this is how the size exclusion chromatography or gel filtration chromatography works based on the principle of size of these proteins. Now, if you have the charge differences, because as you know in the amino acids side chain chemistry, we have some which are positively charged, some are negatively charged. So the overall positive charge or negative charge could be utilized as a way to separate the proteins as well in the ion exchange chromatography, where you can have different type of matrix. One is you have the CM group, carboxymethyl group, or you can have DEAE group, which is diethyl aminoethyl group, which are linked to cellulose or agarose beads. And then when you are passing your proteins are either positively charged or negatively charged. And if in this case you are taking negatively charged resin, they are going to bind, the positively charged proteins are going to bind, and then you are going to desorb them. So after initially you want to get them adsorbed so that they are associated with the positive negative charge interactions. And then you want to desorb them by adding some salt with increasing concentration. So when you add sodium chloride, it is going to compete for this, which is going to remove these tightly bound positively and negatively charged ions. Or you can also change the pH of the buffer, which can also change the charge on the protein, and again the proteins could be eluted out. So depending on if you know the nature of the protein which you are working on, you can choose the right type of resin, and accordingly you can separate the protein in ion exchange. Affinity chromatography is mostly used when you know the specific protein has affinity to some biomolecule, specific substrates, products, or cofactors. Especially when antibodies are being used for this kind of work. And these matrix beads, they are actually chemically coupled to the ligands, and proteins are going to be showing very specific binding through a specific interaction. So in this case, you have this glucose binding protein, which is going to attach to the glucose residues on the beads. Sometimes you are taking a specific antibody, like which is going to really bind to specific protein with that antigen of interest. So many of these things we already know, at least some idea you have that what is the specific interaction you are going to utilize, and accordingly you can set up your affinity chromatography column. And then finally you want to, once the chromatographic affinity has been established, then you want to desorb the your ligand of interest by adding some excess ligand in solution. I'll give you one specific example which is very commonly used for the affinity chromatography, especially in the biolabs. When you want to purify a protein, let's say you want to work on a given protein, you have cloned that gene of interest and you have grown them in the heterologous system like bacteria or yeast, and now you want to take that crude mixture of the protein out, which will have your protein of interest, but also many other bacterial protein and other proteins present in that system. So then you want to specifically locate your protein of interest. So for doing that, even at the talking about in the previous module about the gene sequences and looking at the primers from the PCR. So at that time itself, you are going to add some histidine tag, and these histidine tags are going to be utilized as a way to selectively take out your protein of interest. So now, if the column matrix contains nickel, So now if you're passing your protein of interest, which is having the histidine tag, it is going to bind with the nickel bead. So histidine tag protein is going to show that affinity binding. And then you want to rest everything else is going to pass off. So then you can remove all the bacterial contaminant proteins and other proteins. Wash the column, and then slowly you want to remove your protein of interest, which has already been bound. So red protein, which is the protein of your interest, which is already bound to the nickel beads. You want to selectively move them out. So you can use the imidazole in this case, which can displace this with the nickel ring here, with the histidine binding of that. And then you can increase from 25 millimolar to up to 500 millimolar of imidazole, increase salt concentration so that your protein of interest can be taken out. And you can collect those in different fractions. And you can run each fraction on the gel to find out which fraction looks very specific and pure for the protein of interest which has been eluted out. So again, in this case, you're looking at very specific interactions, which is more utilized for the high degree of purification. Unlike gel filtration, you are not limited with the sample volumes here. And the columns are small columns in this case. In the gel filtration, you use long column because you're using the size-based separation. And this is actually, you are looking at very pure proteins. So this, in the purification scheme of different protein purification, this can be considered as the final step. So that just gives you some idea. I'm sure, as I said, many of you have done much more bigger projects on chromatography. So it was very basic to give you flavor that different chromatographic methods are being used for separating proteins, from gel extrusion to looking at the ion exchange or affinity chromatography. Electrophoresis methods are very heavily used, and after doing electrophoresis separation, you want to separate the protein from the gels, and that comes under gel-based proteomics. So if I'm sure SDSPAGE also has been done by many of you, so which is the single dimensional separation of the protein. The SDSPAGE stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis, where in the presence of the detergent SDS, you are trying to bind that with the protein of interest or all the proteins which is present there, and you are making them negatively charged, and then separating them in the electrophoretic field based on the molecular weight separation. So just this gives you the glimpse of it, that how the SDSPAGE gel looks like, which can be used for determining the subunit composition of the protein or looking at the specific molecular weight. If you know these unknown protein as the standard proteins, then you can say, okay, this could be 60 kilodalton, 70 kilodalton. What is the size of my given protein? You can also look at even if a protein has the 2 phosphate residues attached or 3 phosphate residues attached, and after you remove those phosphate residues, what is the modification? So size can increase. You can determine the PTM as well. And if you don't use the SDSPAGE, but you use the native condition, that is known as blue native page, that can use for the native molecular weight determination. So this was just the overview, but a little bit more detail of electrophoretic and SDSPAGE. This is one of the commonly used technique in protein research in general, and of course even for the preparing the sample and doing the quality checks for the proteomics projects as well. So widely used electrophoretic method which separate protein according to the size. And this is, let's say you have the protein mixture which you want to prepare for. You are going to boil them in the presence of detergent SDS. You also add the thiol agents like beta-mercaptothenol, which is going to reduce the disulfide bonds. And then you are heating them for sufficient time. So idea is you have a globular protein, which is now you are breaking open all the bonds, and you are making them in the linearized form, and then you are making sure that the disulfide bonds are not reoxidized again. So now if the proteins have become linearized and negatively charged because of the SDS which you have added, which has made rod shape, now you can separate these in the electric field just based on their molecular weight. So what is the size, based on that these proteins can separate. So for doing SDSPAGE, you use set of chemicals which you use to make the gels. Those includes acrylamide to make the matrix, bisacrylamide, a cross-linking agent, ammonium persulfate, which initiates the polymerization reaction, timid, which is a free radical stabilizer, also promotes the polymerization. SDS, as I mentioned, is a detergent which makes the protein rod shape and negatively charged. Beta-mercaptothenol breaks open the disulfide bonds. So these are the basics of the SDSPAGE, which is commonly used to just make sure that what protein you have purified or you have extracted, what is the quality of that, what is the submolecular weight size of that protein. But for looking at more of the complex disease biology or looking at actual biological problems of interest, you want to separate really complex mixtures of the protein. So SDSPAGE is only going to give you separation based on one property, which is their molecular weight. But to really resolve the proteins well, scientists also use electrophoretic method, which is known as two-dimensional electrophoresis or 2DE. So here, the first dimensional separation of the entire complex proteome, all the proteins, is based on the pH gradient, because again, given the side chain of these amino acids of the proteins, they will have different isoelectric point, and that could be used to separate them in the isoelectric focusing field in the IEF, which is separates protein in the first dimension. So for doing that, we take some very narrow plastic strip on which acrylamide is coated, and then it contains the pH buffer. So this is known as the IPG, immobilized pH gradient strips, in which now if you put your protein you leave it for overnight so that all the proteins get immobilized as a process known as rehydration. Then you put them in the electrophoretic unit, electric focusing unit, IEF, which separates protein based on the isoelectric point, and that is known as the first dimension separation. Once this part is done, then now you can take that strip and put them in the SDSPAGE, which you already have learned. And now all the proteins can separate based on the molecular weight. So this IPG strip, which was separated based on the PI values, now you're going to further separate in the second dimension based on the SDSPAGE principle, which will separate protein on the molecular weight basis. So all these tiny spots which you see now represents two different properties. One is the PI value, and second is their molecular weight. So every protein we can get right away to information that what is their molecular weight, what is their PI values. A workflow of two-dimensional electrophoresis involves different steps, which we will not talk in detail for this particular course, but just to give you some idea that it involves series of setup experiments, starting from rehydration to the isoelectric focusing as the first dimension separation. Then you want to equilibrate your IPG strip so that your strips are ready for doing the second dimension separation. You actually perform SDSPAGE, and then you want to visualize your proteins on the gels using a different type of staining methods like Kumasi blue stain or silver stain. You want to analyze the images. Image processing softwares are there which can be used to do the image process analysis. Spot picking to identify which spots are significantly different. So again, you have to do a lot of statistics to make sure that which of the protein among thousands of spots are showing the common pattern and showing good p-value. And then you can pick up those and digest those and want to know what those proteins are. And then you want to do the mass spectrometry based analysis for that. So IEF, conceptually how this works. So you are taking your IPG strip, and let's say in this case we are taking 3 to 10 pH gradient IPG strip. Wherever the pH becomes equal to PI value, there is no net charge, and that migration will stop. And accordingly, all the proteins are going to move in this particular isoelectric focusing unit. And depending on what gradient you have provided, 3 to 10 pH or 4 to 7 pH, in that pH range, all the proteins will move to respective PI values, and you can separate them based on the first dimension IEF. So for doing that, these are the kind of instrument setups which we have, which we can use for this experiment. Here you can set up different voltage so that voltage is stepped up. Slowly you increase the voltage because all the protein has to focus, but you can't just provide too much voltage to begin with. So you start with 200 volt, 500 volts, slowly ramp it up to 1000, 8000, and keep it static at that so that the volt hour could be reached. And you set up the volt hour depending on the length of the strips, the amount of protein which you are loading. And accordingly, you can set up a 50,000 volt hour or 80,000 volt hour. And you want to achieve the blue trajectory here for the IEF to happen. But sometimes you see these kind of dips happen, which could be impurity of the sample or the salt available in the sample. And then you have to do troubleshooting to find out how to overcome this problem and change the weights or the IPG strip and do another round of experiment. So these things are part of this experiment. If it goes well, then you will take these IPG strip and dip them into solution, which is DTT, dithiothriotol, which can do the reduction. and followed by you dip into the second solution, which is the hydroacetylamide. So now what you want to do, you want to break the disulfide bonds using DTT, and then you want to alkylate with the hydroacetylamide so that these are forming the sulfhydryl groups, and they're not going to reform and do the reoxidation. So you want to prevent that so that proteins are still remain to be denatured, and they're ready to be linear to separate in the second dimension SDSPAGE separation. So again, to just summarize, that we have done separation in the first dimension using PI values in the EF unit, second dimension with the SDSPAGE. Every spot denotes their molecular weight and PI value. And same principle has been used for many biological projects, different type of clinical problems, to look at all the protein expression in a given system, to compare the healthy versus disease comparison. If you have a complex mixture, which is you have pulled down from a given antibody, you want to identify their immune precipitation, you can look at that, or look at different isoform of the protein or PTM modification. And all of this can be done by looking at even visual analysis of the gel, because you can stain your gel from variety of staining methods. Kumasi stain is one of the commonly being used and very cheap stain available, quite stable. But you have also other options like silver stain, cypro-ruby, cypro-orange, cyanine dyes, lightning fast, Proteome, many stains, and I have shown here their sensitivity level. Some of them are quite sensitive, like silver stain and cypro-ruby. Silver stain had some compatibility issue with mass spectrometer. Cypro-ruby is quite friendly, so it can be used for a variety of application. Kumasi brilliant blue, that is one of the most commonly used staining method, which you see in most of the molecular biology labs. It is very stable. It forms ionic and non-covalent interaction with the proteins. So it is once it binds with the protein, you can then do the destaining, and then those stained part will not go off. So as a result, you can see very less background, and only the proteins will be more visible, which will be blue colored. And you can just keep it overnight in the stain, and then next day you change the stain solution and keep the destain. So as a result, this is one of the very commonly being used staining method. But if you're looking at more sensitive stains, and also if you want to do multiple type of experiments on the same gel, you can use the dual staining method. So for instance, you are looking at a specific type of modification like phosphorylation. So you can first use this Proteome stain. So you stain your gel with Proteome and then obtain an image with the 580 nanometer phosphoprotein imager, and then destain that overnight with just simply water. And after that, you can stain them with the cypro-ruby red, another stain, and again take that measurement at 610 nanometers. So entire gel you are scanning. So same gel, now you are able to obtain two different images of phosphoprotein and the total protein, and then you can see which proteins are also getting modified. So one could do different applications in this manner. To take this technology forward, so what we are trying to also give you idea that how to do the basic biochemistry type of experiments and take it forward to more proteomics type of high throughput experiments. So SDSPAGE, moving on to 2D gels, and then now let's look at the dye-etch technology difference in electrophoresis. So in this technique, now we want to compare, let's say our in the COVID project, we want to compare some individuals are having mild fever and some individuals are having severe fever or severe symptoms of COVID. And those two are very different. Like, one is definitely going to recover, one may recover, may not recover, right? So you want to know that what is happening in the mild, which is different than the severe group of patients. So now you got interesting problem to investigate, and you have two set of patients. One is mild, other is severe group, or you can say non-severe and severe group. So now you take, let's say, blood sample from each group, and to make a simple problem, let's take 5 individuals of non-severe group of COVID-19 and 5 patients of the severe group of COVID-19. We took the blood from them, plasma from them, extract the protein, and then you want to label the proteins from the the mild group with one dye, which is, let's say, Cy3 dye, and other group you want to label them with another dye, which is Cy5 dye. Now these dyes, they are quite photostable, pH insensitive, spectrally distinct. Chemically, they are formed from the NHS ester derivatives of cyanine dyes, And I have provided some detail on the structured side and the excitation emission wavelength. What is important for you to remember here that both size and charge wise they are matched. So if let's say you are going to take one patient which is non-severe, one patient severe, label that sample with Psi3, label this sample with Psi5, and then you mix these two together and then run them on one gel. So they are going to migrate to the same, same size and same charges matched already. So then you will not see any effect of the dyes, which is, the sample was labeled with Psi3 or Psi5. Likewise, you can do the same pairing for all the 5 sets which we talked, and then you generate these 5 images of the gels, and then you can do comparison of these images to find out what is happening in these patients. I will talk about this experiment as I go along. So again, for these type of technology to develop, a lot of chemistry has to happen, a lot of new dyes has to come forward, and the field is still limited with the how many dyes which we can have for doing multiplexing capability. So again, we have a limited number of dyes, cyanine dyes, only 2 are most reliable, like Psi3, Psi5, and Psi2 is another dye which is also used for some internal standard comparison. So in these cases, we have the protein will having the amino terminal. You are going to target that for this particular Psi dye to bind to that. But for doing this particular reaction, you are looking at very specific condition at 8.5 pH, and you're looking at the cold condition and 30 minutes of incubation, so that then the dye and protein can bind, and that can on a very specific actually amino acid which is lysine, this particular binding is actually happening, and it forms the covalent amide bond. So if this binding has happened well, now you can think about the experiment which I'm talking to you, that you have some set of individuals which are, let's say, mild patients, some set of individuals which are severe patients. So first you have extracted the protein from each one of those, and then you took, let's say, 100 microgram protein from each one of those and mix it together, and that you made a pool which is now representing all the proteins present in all the patients. So you will keep your entire protein separately for each patient anyways, but still you are making a pool of the protein so that you can have some sort of benchmark as a common fingerprint for every protein present in all the given sample. And this one you can use to label with the third dye, which is Psi2 dye, cyanine 2 dye. So a typical experiment, now we are talking about interesting COVID experiment, where we have some group which is mild, some group which is severe, and we have a pool of all of them, all the sample, which we have labeled with third dye, which is Psi2 dye. So then all the three, once we have done the labeling, which I've shown you the labeling reaction, we can mix all the labels together and then separate them in the IEF on the IPG strip, followed by on the 2D gel, and then obtain these images from the same gel. So now Psi3, Psi5 represents image of one mild and one severe patient, and Psi2 represents image for the internal standard, which is represent the pool of all the 10 or all the 8, whatever we are comparing here. But likewise, you can have second gel, which will give you image for the second patient mild and second patient severe, third image and fourth image, and likewise. But this image will remain same. The Psi2 image will remain same because that is what you are going to use for internal standard comparison for the analysis purpose. So earlier we had done some projects using this technology, which was on the malaria. We just recently published a paper in the Nature Communication Biology, which is on the malaria severity. So we were looking at some malarial protein, how they are different in different type of infection of malaria with falciparum and vivax, and it just showed that some of the protein which looked different from healthy individual to the different disease individual for different proteins. And also the same protein, when we looked at in dengue fever, it was not showing many response. It showed really huge response in the 2 malarial fever like falciparum and vivax. So based on these experiments, this is kind of interesting observation you can make. And then you want to go and find out what this protein is, which is showing me this kind of sharp 3-dimensional peak. And then you can use the mass spectrometry technology to identify the proteins of interest. So the kind of experiment which I'm talking to you involves protein extraction, protein separation from 2 different conditions. Then you are separating them in the isoelectric focusing unit, doing reduction alkylation, separating them on the either 2 gels with the SDSPAGE or 1 gel with the 2D dye, and then compare the images, excise the spots, and take those forward for mass spectrometry based analysis. So that brings us to really complex technologies now, which is I just showed you our laboratory views and the setup here, where we can now use these type of high throughput gadgets to identify the proteins in really high throughput manner. So mass spectrometry is one of the technique to do protein identification and analysis by producing the charged molecular species in vacuum and separating them in the magnetic and electric field based on mass to charge ratio. So unlike the standard balance which you are using to weigh your compounds just to look for the mass, here you are looking at the various ions and their mass to charge ratios and going to use that information for further deriving your peptide sequence and use that for database search. So broadly, when as I showed you earlier that you can put your sample in the nano LC or UHPLC, that is the sample inlet, which is the liquid chromatography part. Then from there, the ionization source comes, which is the, let's say, electrospray ionization, where you can ionize your protein in solution form or peptide in solution form to now change to the gaseous ionized forms. Ions you are separating in the mass analyzers, which is based on the mass and charge ratios. And then that reaches to detector, which is going to establish the spectrum, which I showed you in one of the screen over there, that how this chromatograms looks like, which is showing the separation of the peptides. And then you use this particular data set to search for the databases and do the data processing. So one of the complex term, which is known as the shotgun proteomics, is essentially that you are like, you are injecting your complex protein mixture, which is in a tube. You are not separating it on the gel the way I showed you in the last module of the two-dimensional electrophoresis, but rather you are directly subjecting them to the mass spectrometer. And for doing that, you are taking the entire protein mixture in a tube. You are doing the, with the maybe urea denaturation, followed by reduction with DTT, alkylation with IAA, iodostamide, and then you do digestion with the trypsin or chymotrypsin or lysine enzymes. Followed by you got now these peptides, which you are cleaning up for removing the salt, and then run them in the mass spectrometers, like when I showed you Orbitrap, but there are different type of other mass spectrometers available as well. And then you can proceed for the data analysis part. So any mass spectrometer, I think it's a good idea to spend a little bit time on mass spectrometer. This is currently, really important technology which can do a lot of different analysis of proteins in different way. So there are three major components. One is ionization source, second is mass analyzer, and third is detector. Additionally, you also have three other accessory components. One is sample inlet, which is the liquid chromatography or how you inject the sample, instrument control, data processing setup, and data output, which you again going to use with the data bases and software analysis. So there are different type of mass spectrometer configuration available. So one of the popular configuration is multi-TOF. So multi-ionization source matrix assisted laser desorption ionization, and TOF time of flight is a mass analyzer. So in this setup, let's say you have a in the multi, and this is the this part is the entire time of flight, and you have a plate on which you are going to keep your protein of interest, and that protein you are going to mix with some matrix, some organic reagents which can make the crystals, and now these crystals you are going to bombard with a laser beam here. And after bombardment, now these ions are formed from your protein or peptide, whatever you want to analyze, and going to move it in the time of flight tube. And then what you're looking at, how much time it takes for these ions to fly in the analyzer and strike and reach to the detector. So the mass discrimination you are doing based on the flight time, and they are exponentially proportional to the flight time, how much time they take to reach to the detector. And ions of the lower masses, they are accelerated to the higher velocities. So based on how much time they take to fly in the TOF tube, you are separating these ions based on the mass and charge ratio. And you can now think about additional complexity of the instrument, which is multi-TOF. So in addition to one TOF, which we just talked, you have another TOF tube as well. So now what we have is a multi as an ionization source, followed by a TOF tube and followed by another TOF tube. In between the two, we have also a collision cell, and collision cell is essentially which contains some of the inert gases. You can have argon, nitrogen, these gases. So what is happening when you are taking your protein mixture, which is on a plate, multi-plate, which is mixed with the matrix? You are doing laser bombardment, ionizing this. Now these peptides or proteins are moving it in the field, and now they are getting separated in the top one based on the time to take how much time to fly in the tube. And then most abundant one you are defining to the software that pick up maybe top 5 or top 10. And so let's say it picks up one of the first most abundant one, and in the collision cell with the inert gases it does the with the collision energy it does bombardment, and then now generates further fragmentation of that one most abundant ion, and then picks up the next ion, and then excludes that over there, and then again does the bombardment and generates the MS spectrum of that. So what you generate the data here, that is the MS data only. You're only looking at mass. And when you generate here, that is MS, you are generating the peptide sequences, because from this data you can derive the peptide sequences. In addition to these, the detail which I'm talking to you, important thing here is the reflectron mirror, which is part of these TOF tubes. So, to really separate these ions well, you want to have the much longer path length for the TOF tubes. They used to be very tall, 2 floor long TOF tubes, but now, with this kind of reflectron mirror, you can increase the path length, and that is now part of these multi-TOF instruments, which can be reasonably still big for a room size, but need not to break open the wall. Another type of mass spectrometer is based on the quadrupole technology, quadrupole mass spectrometers. Here you have a set of these rods, which is the parallel metallic rods, and ions are going to resolve based on the oscillating electric field. So you change the voltage here, and by varying the voltage over the time, then you are going to separate based on the mass and charge ratios. And this particular quadrupole mass analyzer is also independently used in the mass spec, as known as triple quadrupole mass spectrometer, or it can be interfaced with other type of mass spectrometer like Q-TOF. So you can have one quadrupole, and along with that you can have the TOF, which you just talked. So popular mass spectrometry configuration, where now you're utilizing 2 different properties. One is mass analyzer quadrupole, other is the TOF, time of flight. So you can first separate them based on the electric field, select those ions, and then you can further separate them based on the time of flight for the path length. So these are different physical principles, which I'm sure you're very comfortable now understanding, that being used to separate these complex ions. So now to take a minute back and give you a bigger picture, what we are talking in a tube, we have 10,000 proteins, and that 10,000 protein we have digested with enzyme like trypsin. We got now maybe 50,000 or a million peptides. Those peptides are in liquid form, and we are injecting from liquid chromatography slowly, and they are now getting converted to the gaseous ionized form using electrospray ionization. And from there, they are now coming to this mass analyzers, like different type of mass analyzer, which I just showed you. And then you are trying to separate those complex ions, which are actually nothing else but going to give you a peptide sequence or protein sequence eventually. So then you don't want to lose them. You want to read as many as possible, and that's where it's your resolution and your sensitivity is going to be determined. So one more recent addition to this type of mass analyzers is Orbitrap, which is really high resolution. It is based on the ion trap principle, which is going to really take some of the ions in this type of trapping field and going to separate them in the Orbitraps. One technology which I showed you just now in our lab is actually Orbitrap fusion, which contains tribrid type of mass analyzers. So in that case, we have a quadrupole, we have ion trap, and we have the Orbitrap. So 3 different type of ion trap is here, ion trap and Orbitrap. So 3 different type of mass analyzers are used. So what you can think about now, in addition to just thinking about one type of mass analyzer to separate the masses, one could also use the hybrid configuration like Q-TOF, 2 different type of mass analyzer, or tribrid type of mass spectrometer like Orbitrap fusion in this case. What it gives you ability, it gives you on one hand more ions can be kept on hold. Slowly they can be taken to different ways for separation with ion trap and Orbitrap. It also gives you more type of collision ability with the HCD and different type of high collision density, which you can do dissociation of these ions. And overall what it gives you more ability to increase the resolution. So Q-TOF can give you limited resolution of 30 to 40,000 maximum. In Orbitrap that can give you 1 lakh to 5 lakhs, that kind of number. So, but the basic principle here is that whenever we are taking these ions, which we want to further use for our peptide spectra or protein spectra to determine, we are telling the software that whatever the most intense precursor ions is coming, you first select that and isolate that in collision cell, which I just told you. Between TOF and TOF, there was a collision cell. Between Q and TOF, there is a collision cell. So now this collision cell is going to use to take the most intense precursor. You are going to do collision induced dissociation of that and generate these kind of fragmentation spectrum with the MSMS pattern. So this is what is most important, and that's where when I was talking to you about peptides and amino acid, I gave you this brief idea. I'm again repeating this again here. Now you are much more clear what we are talking. So what we are looking at in these kind of collision cell, we are looking at now these how to generate these peptide sequences. So now we have, let's say this is amino terminal and carboxy terminal. We have multiple peptide bonds. So with the low collision energy, these peptide bonds are going to get fragmented, and the part which retains toward the carboxy side is termed with the Y ions. The part which retains toward the amino terminal terms as a B ions. So for any of the fragmentation to happen, you will always have complementary ions from B and Y ions. If it is closer to N terminal, it is the B1, closer to C terminal, it is Y1, but you will have corresponding series of both B and Y ions. There is a software which is used to search this kind of mass spectrometry database, which is open access, that is known as Mascot. Here you can take this mass spec data and upload that with some parameter to be used, and then you can see what that protein is from the given sample which you really wanted to analyze, or you can have maybe multiple proteins which you wanted to analyze. In this case, let's say you got one of the heat shock protein, and you got many peptides which is used for searching this protein in database. So your mass spec has determined these peptide sequences, and now these peptide sequences, how they came forward, how you are able to get these peptide sequences. That's where I think the basic concept which I talked to you about B and Y ions becomes very crucial. The differences of these Y ions are representing one amino acid. So Y7 minus Y6 gives you 57, which is characteristic of glycine. Y6 minus Y5 is giving you a difference of 129 of glutamic acid. And now if you look at the corresponding B ion series, that is giving you still same difference because the B and Y ions are complementary from opposite sides. So that confirms that this is actually right amino acid. And this is what software does for us. It quickly generates that peptide sequence from that spectra. So I hope now you are able to correlate, where I'm trying to give you different concepts and why trying to correlate that how peptide sequences are important, and using mass spec you derive these peptide sequences based on the B and Y ions and look at those to find out what the peptide sequences you have. Then you can, if I just go back here, then you can use that information to search the databases, and that will tell you that what this protein is, right? So I will give you actually maybe some data set as shortly, and you can try to do the search yourself, which will be very helpful for you to get more comfort of doing this kind of analysis, which is still not very big data and high throughput data, but at least some basic set. So I'm going to give you one tutorial and some data set to do some practice on that. But I hope conceptually it is now getting much more clearer. Now this part is slightly more complex slide. Please do not worry if you do not go through completely, but this is what is the most recent thing happening in the proteomics field. When you want to compare, let's say now we are talking about same 5 of the non-severe and 5 of the severe patients of COVID, and we want to compare all their proteins which is changing in their plasma samples. So now there could be different methods as we talked. You can use the 2D gels, you can use dye gels, but they are more cumbersome to do those kind of experiments. Also, they are going to give you limited data set of as maybe a couple of 100 proteins you can identify. But using mass spectrometer, and especially more sensitive and high resolution mass spectrometer, you can really generate thousands of protein and very high quality data in a very short time. So for doing this kind of work, there are different approaches which people are using in mass spectrometry field, which is some of these complex terminologies are there, which is one is known as DDA or data dependent acquisition, other is known as data independent acquisition or DIA. Again, just think about the basic concept. We are looking at how to select those ions, which is the most intense precursors, and how to fragment those. All the tricks are lying over there. So either you select the single precursor, the most intense one, fragment those, or you select multiple precursors at the same time and then do their fragmentation and detect all the fragmentation ions. So this is what the difference is between data dependent acquisition and data independent acquisition. But if you have already known that which of the protein or peptides are of your interest, which you want to now do for testing of COVID patients, then you can set up these type of tests, which is known as multiple reaction monitoring or parallel reaction monitoring, where you're only going to selectively look at those specific peptides and want to monitor those peptides and see like are their levels are changing in the mild versus severe patients. So again, I'm not going in detail on this slide. I still wanted to give you flavor. This is the most recent thing happening in the proteomics field. If any one of you have more interest, I have actually full lectures and a lot of details on these technologies. We can give you more information further. Other technologies and approaches which is commonly used in the field of proteomics, you have got some flavor of it, especially mass spectrometry based from mass spec to protein microarray based approaches. And microarray, you have seen Professor Graham Ball giving you some data from the Array Express and some analysis of that. So we also use the array-based platform, and the applications are different. Sometime when you are looking at autoantibody type of screening or protein-protein interactions, for those kind of tests, you have the microarray, which is very powerful approach. But many times when you are looking for many unknown proteins and large number of protein separation, mass spectrometry can be much better and a strong platform. So there are different type of arrays which one could use. If you have availability of antibodies, some of these array platform could be used known as abundance-based arrays. If you have purified proteins, you can use some of the functional-based arrays. Many times in the clinical settings, the clinician or pathologist, they want to use reverse phase arrays. They can take the, let's say, patient's tumor biopsies, put that on the chip, and now you incubate that with an antibody. Let's say for PSA, you are looking at a man as having prostate cancer or not. So you are taking the antibody, which is anti-PSA, prostate specific antigen antibody, and then if that binds to that patient sample, it means that patient has expression of that particular protein, and this is how these kind of array can be used for quick diagnosis of these disease. Once you have identified some proteins of interest and now you want to develop some drug target and see whether the drug is going to bind to these proteins or not, then you have the more biosensor platforms like surface plasmon resonance or biolayer interferometry or other type of biosensors which are conductance based or reflectance based. Many of these are available. This is reflectance based when you have a prism, a light source, and you have a gold slide, and you are looking at the change in the refractive index of the medium or percentage reflectivity change. This is an antibody, let's say, immobilized on the gold slide. A protein is coming which you are flowing and you want to see the binding is happening or not. If the binding is happening, then you are going to slowly add the buffer, and over the time that binding will get removed. So these molecules are going to come out of this, and you can monitor that in this kind of chromatograms and this kind of sensorogram, where you can see on the time versus response unit scale. Initially there is no binding, baseline is static. Then some binding is happening, that is an on rate, which you can see for this binding to happen. Then once you start injecting more buffer, that binding is getting dissociated, that is known as dissociation constant or the off rate. And then you want to regenerate that chip for the next cycle. So you can use some mild acid to remove any of the bound material and then wash them again and use for the next cycle. So again, any of these, we have a lot of content material and the hands-on sessions available. Please feel free to get in touch with us if in case you are interested in any of this specific technology which I have been talking. I'm just trying to give you a flavor right now. I can't go in great detail for every technology which I'm talking, but we can definitely talk to you in much more detail about any of these. And also we have a lot of lab sessions which we can share with you. And we are also free to take any of the samples which people want to analyze in our facilities, in our instruments, and also want to collaborate and want to utilize some of these resources which is given by government and set up at IIT Bombay. So we will be very open to take these interactions and knowledge and learning forward. Now coming to the realistic situation, when we talk about any project, the project does not involve just only microarrays or only mass spec or only SPR or only chromatography. Any type of project actually eventually involves all of these technologies. So when we talk about a simple project like, let's say in this case COVID, and we are having blood samples which we want to analyze, you are doing the protein extraction, doing the mass spectrometry analysis, finding the PMF and MSMS analysis to find out which are all proteins present from the virus as well as the human side and want to analyze those with the databases. Or you can take not the tissue biopsy in this case, or even the autopsy sample. People are actually analyzing right now those who are dying patients, even their some of the biopsy for different organs have been taken now, and people are analyzing what is the impact of that on lung and other kind of organs in human. But that we do mainly tissue analysis in the cancer projects. But nevertheless, in this case as well, you can use even microarray type of platform to look at specific interactions or specific binding with the drug, or you can look at even the autoantibody type of markers. In case of COVID also, it's quite relevant that we are looking at the IgG, IgM, these kind of immunoglobulin responses. And if we can have these viral peptides on the arrays, we can map the epitopes, that which epitopes are showing the maximum humoral response for with the serum if you take from a patient samples. And some of these experiments are actually going to open up more and more knowledge, new knowledge, which is really required right now to really understand that how this virus is working and what more options we can have available. If this vaccine does not work, do we have more epitopes, multi-epitopic regions which we can target, and we can come up with a new vaccine in subsequent timeline. So all this is really important, and I'm just giving you one example right now, but the same way any kind of biological project or clinical project could be heavily benefited by using these type of technologies. So just to summarize, while there are many technologies which have really been important, but mass spectrometry is really high throughput and become one of the very crucial technology. Of course, understanding some of these basic terminologies and techniques are very crucial for anyone who want to utilize them in their research. But especially different type of hybrid and tribrid mass spectrometers, they are quite important for doing any research. I hope I'm able to give you at least some flavor. I have not intimidated you that these are very complex technologies. At least give you some flavor, some excitement that the basic chromatography, basic electrophoresis, along with some of the newer additional high throughput approaches like microarrays and mass spectrometry can make you really a good arsenal, various tool which you can utilize to really combat towards different type of infectious disease or different type of metabolic disorders like cancer, and try to provide at least some better understanding or better solution to fight against these real, sometimes invisible enemies, and sometimes the visible tumor growth which you can see. I think that's all from my side. Thank you for staying in all these sessions, and I'm very much open for interaction and discussions now. All right, so we are here in one of the Proteomics mass spectrometry lab, where we have set up various high-end mass spec instruments, which we are going to use for different projects. Of course, you are going to see them as I talk to them in more detail. Just to give you some visual sense of this, that what we are talking. We have been talking about analyzing proteins and complex protein mixtures. Those could be processed here with the liquid chromatography interface here. And if you could just, I hope we can open the screen. Yeah, so some of these complex samples, we actually bring it here, and then we try to process them with the liquid chromatography, which is nano LC. Then we are trying to use different solvent system with the actonitrile formic acid gradient. And then we have these columns, which is rich in the C18 material, reverse phase column, where all the peptides are going to come from here, and then they are going to reach here. Then they are going to, as you can see, then set here. They are going to move from this part with electrospray ionization, and then go to the mass analyzers. So this is actually the black box, which is really the big place when you can separate all these mass to charge ion species. And what we see here is one of the orbitrap fusion tribrid instrument mass spectrometer, which is having quadrupole, orbitrap, ion trap, 3 different mass analyzers to provide very high resolution for doing protein-based work and proteomics research. On the screen, we are just showing you one chromatogram, which is essentially, if you can look into what I'm trying to show you on the TV screen, that shows how the peptide spectrum looks like when we run in the mass spec. Of course, we will show you some actual data sets. Can you show a Dell's chromatogram? Yeah, so what you see that the peptides are separated in 2 hours time with a gradient, and those provides sufficient opportunity for the complex peptide mixture to separate from the instruments. Uh, this is another type of mass spectrometer, which is Q-scattering mass spectrometer, and which is linked with the UHPLC, which we are using for generating an additional data with the metabolite level, metabolomics level. So this is the metabolomics data set which we generate here, and additionally one could also generate specific peptide sequences or protein sequences data, look at targeted validation of peptides that we monitor in this instrument, which is the triple quadrupole uh, mass spectrometer. Of course, I talked to you about next generation sequencer quite a bit. So those instruments are here where you can do some NGS based assay. So these are, as I said, these are the workhorses of generating a lot of big data, and one could do complex protein mixture analysis, proteome analysis from this. I think my this video was not visible earlier, so I'll just show you quickly the lab glimpse if it was not seen earlier. So again, in the molecular biology lab, you have variety of basic technology which you can do for doing the DNA-based work and the protein-based work, and those could be used to investigate really complex problems. And we do a lot of work in the different clinical projects, especially brain tumors and some infectious disease. Right now, we are doing some work on COVID as well. And I was trying to show you that, maybe just if you can open one of the freezers, how these samples are really stored. They're stored in really cold condition of almost -80. And you can see so much, every box contains these clinical samples, or we contain some of the genes of interest clones in the glycerol stalks. So these could be used for variety of research, and but their storage, their way of processing, all requires various workflows, SOPs, and quite a bit of optimization which we do in the lab, right? So this is a typical bio lab and molecular biology lab, but more than that, we have a lot of omic technologies and mass spectrometry, proteomics, microarray based proteomics, which generates big data sets, and those big data sets we then process from different clusters and servers which we have. But even sometimes they're not sufficient, so we have to use some cloud space as well, and use of collaborative space with the other projects which we are doing with international groups. So I hope this is giving you at least some excitement that how many of the basic concepts which we are talking will be eventually utilized to do good research for